Hi, hello everyone. Welcome back to my YouTube channel, My Intuition 4865. Continuing my classes on biology for engineers, that is with the subject code 21 BE 45, which is common to all the fourth semester branches of engineering branches of Visvesvaraya Technology University, VTU, Karnataka. Previous class I started with chapter 1 biomolecules and their applications, in which I discussed about carbohydrates. Now this point 2. Okay, so nucleic acid. Nucleic acid, they are a type of polymers. Polymers means very long chain molecule. But to our eyes, they are still not visible. Not see that they are just like a rope and they are long chain molecules compared to the other small molecule. They are biopolymers. They are bio means they are biological originated polymer structure, which play a crucial role in the storage and transfer of genetic information in all living organisms. That means from generations to recall the generation, the character, the nature are getting engrossed, or from our new generation, from our previous generation, new generation, they are more advanced because already what we learned, that is there in our gene, that we are transferring to our next generation. Therefore, the information already there, they need to just brush it up and collect more information. Therefore, these gene informations are also transferred, all the character, all the behavior, nature, all that talents, everything are transferred to the next generation. There are two types of nucleic acid. That is when I say about genetic gene, I said something about that. Deoxyribonucleic acid, DNA, as well as ribonucleic acid. These are the two different type of nucleic acid that we already studied. DNA, because the D represents deoxyribose. The base structure, it consists of deoxyribose molecule, and also DNA contains the phosphate groups, nitrogen base like adenine, guanine, cytosine, and thymine. Ribonucleic acid, that is a single stranded DNA. It is a helical structure, double stranded helix structure. Okay, so the difference only that RNA is single stranded, and in place of thymine, the nitrogenous base thymine, the RNA molecule is having uracil. Okay, so that is a difference. They all do almost the same function. DNA, that is it is the genetical information. It carries the, it acts as a memory device also in our body system. RNA, that is used, it is working as a copying material, copy to copy paste, to help generation of new proteins, which is required for our body whenever it is required. So the structure of both the schematic representation of DNA and RNA is given some color formation just to understand. They are not like a thread or a rod like that. This is just a schematic representation only to imagine how it will look like when we go to that size level. We cannot go to that size level, but you can imagine there it is like that. This type of possibly this type of, not so exactly, because still I'm not sure that our intelligence reached to that level that we can fix this is the structure. Still it look like should look like this one, so that it giving the different functions that we are facing. So both DNA and RNA, they play essential role in the functioning of the cell and the organism. Their structures, the interaction with other molecules are the basis for many biological processes such as the replication, transcription, translation. Replication means multiplying the cells. Transcription means reading the genetic code. Translation means creating new proteins or something. Okay, so I'm not going in detail about that. Okay, not required for all engineers, only this basic information required. Why DNA is so important now nowadays? DNA is always important because it is also producing vaccines. Many diseases like cancer, rabies, influenza, HIV, etc., or can be cured with the vaccines which are produced by the DNA of the virus or the bacteria which causes that particular disease. So we have to take the piece of that virus or bacterial DNA we have to take. We have to extract the pure form of that DNA. We have to put into the body cell bacteria. Then the proteins will be generated. More proteins will be generated, and these proteins are displayed on our surface of a cell. Then the cell will automatically try to react to that. It will try to overcome that, then produces the antibodies, so called. So whatever new thing which comes, new infecting molecules, pathogens, or microbes, when it enters to our body will create antibodies. Our DNA, RNA, they produce by using the amino acid molecule present in our body. It will produce a weapon, a protein, or something which we call as the antibodies. The antibodies will go and kill that virus, or it will cover the virus without creating any harm to our body. So this is generated separately. These antigens are generated by using the viral or bacterial DNA. Using that, it is separately generated. These antibodies are generated. These antibodies we call as the vaccines. Okay, so these vaccines are used for the treatment of cancer, or rabies in mainly in dogs, then influenza, then HIV, human immunodeficiency virus. Still the technology, it is improving. More and more research need to be done, and we can cure almost all diseases. Our body is capable of curing any disease, but we are supporting it by producing it separately and injecting, than waiting for our body to generate all that antibodies. We are creating some antibodies separately and using it. That is helping us, but it should not help us. Actually, our body should have that potential to generate all the antibodies required. Whatever microbes comes up, our body should be able to produce the antibody to resist that, so that our normal functioning, biological functioning will happen, than getting ill. Okay, so for rabies, DNA vaccines are used for rabies. Again, it is taken from the rabies virus. DNA is taken from the rabies virus. Then it is injected to the body, where it forms the proteins. More proteins are formed. Then it is injected to surface of a cell. Then immune response and antibodies are formed. Same sentence. Okay, so importance. So what are the importance of DNA vaccines for rabies? Some of the, like, it's having very good efficiency, high effective in preventing rabies infection in both animals and human trials. One study say that vaccine found to be effective as a traditional vaccine in protecting dogs and dogs against rabies. It gives a long-lasting protection. It is very easy to administrate, very easy to handle it, cost effective, little bit relatively inexpensive. There is no side effect, reduced risk of side effect, because it does not contain any living virus particle. Therefore, further side effects are reduced. RNA vaccines. RNA vaccines are a type of vaccine which uses the genetic material from a pathogen in the form of a RNA. Okay, again similar to that of a DNA, how it is working, same type RNA vaccines also work. They have several advantages over traditional vaccine, because they have very faster production time, ability to target multiple antigens, and the vaccine can be manufactured quickly, making them a good option for emergency situations where large number of people need to be vaccinated quickly. So fast production, the factory is very fast producing. Okay, RNA vaccines are currently being developed and tested for a range of diseases including COVID-19, influenza, and cancer. Okay, so side heading in your syllabus, importance is given for COVID-19. COVID-19, to control the COVID-19, initially we used the RNA vaccines only. RNA vaccines produced, the first RNA vaccine for COVID-19 was produced by Pfizer Pfizer Biotech vaccines, which has been authorized in December 2020, and it was administered to millions of people over the world. Then another RNA vaccine, that is Moderna vaccine, were also authorized in December 2020, and there are many other vaccines are produced after that. What is the importance of RNA vaccine for COVID-19? They having high efficiency. The rate of clinical trial it reached up to 95%. It means 95% people are cured, or they are resistant, become resistant to the COVID attack, SARS-CoV virus attack. Then rapid development. Under a year, within a year, it has been developed. Uh, it has been get authorized within 11 months. So therefore, rapid, whenever a crisis comes, so we can immediately can think about RNA vaccines of that particular virus or a particular microbe. It is easy to modify. We can do some modification, as the we know that we heard that COVID-19 it getting converted, different forms are mutation is happening, and different forms are getting generated. Then we also will mutate our RNA vaccine. We also do accordingly. We do the changes. That is easy to modify. The possibilities are very high in case of RNA vaccine. It is very easy, safe to use. It provides proper protection from virus. Potential for broader use, used in the prevention of other infections such as influenza, HIV, Zika, the treatment of cancer, etc. DNA fingerprinting, in case of forensic, DNA fingerprinting is used for many other applications, concentrating only on forensic, that is criminal related investigations, that is called as forensic. It is also known as DNA profiling or genetic fingerprinting, which is a technique used in forensic science to identify an individual based on the unique DNA profile. Every individual in the world are unique because of their DNA. Okay, we use our fingerprint to uh, identify us. Same way, our DNA is also a unique one. Our fingerprint, that pattern that we have, that is individually, every individual has got their own pattern, and it cannot be copied as such. Same way, DNA individuals are having their own DNA, even though we say that genetic information is passed to next one, but there will be some modification there. It is not the same DNA, because there is a male female when combined, then only a new this life is born, new life is formed. So whatever that individual, their DNA is different only. Only thing that we have to take our DNA, we have to replicate it, and we have to make our own copy. That is possible. Then DNA will be matching. That is, the government are not giving permission for humans. Animals trials has been done, and you know that some of the examples are also there in which exact copy of an animal is produced. I'm not even naming that animal also. Okay, don't think about that, leave it. We can think about DNA profiling, forensics. Working of DNA fingerprinting for forensic application, how does it work? You have to collect our DNA sample uh, in the form of any blood or semen or hair, any some body part. If it get it is possible to separate the DNA from that. Then it has to be amplified. One small DNA strand if we get also, we can copy it, make so many copies with a process known as polymer chain reaction, called as PCR. We might have heard about RT-PCR during the COVID-19. That PCR is polymer chain reaction. PCR produces many copies of a specific DNA region, which allows for more accurate analysis. Then analysis, amplified DNA is then analyzed using a technique called gel electrophoresis. The DNA fragments are separated based on the size and the charge, and the DNA profile is generated. Okay, so different DNA, they having different size, length, size. They having different charge based on the carbon, hydrogen, oxygen they present. Everything has got its own energy levels. So the different charges are there. There are different charges will be there. Based on that, DNA profiling is done. Then it is compared with other individuals such as the victims or other to get a match. Once it matches, then that is exactly the proof that forensic application is done. Okay, so DNA comparison typically done manually by forensic analysis. Nowadays, with the introduction of artificial intelligence (AI) algorithms, the data science improvement, AIDS, as well as with that artificial intelligence, so many data are already available. Artificial intelligence will try to automate this DNA profiling system, then it will be faster compared to the present system. So the DNA profiling consists of series of bands on a gel which represent specific DNA fragment. The bands are compared to those from the controlled samples such as blood or saliva from the suspect or victim to see if there is a match. If there is a match, it is considered strong evidence that the biological samples came from that individual. That means already the one who fought for any criminal case, the forensic expert will collect their DNA sample and keep the copy in their system. Okay, then they will whenever the crime happens, they will take the sample from the victim. Then they will try to match whether in the list if it is already there or not. If not there, then all the suspects when a crime happens, all the suspects DNA also will be taken, and then it is compared, and if it matches, then that gives the result. Okay, so that is about DNA fingerprinting and that forensic applications. Okay, so this DNA fingerprinting, it is already used for many other applications also, to identify any argument that the kid belongs to whom. All those different missing person, identify a missing person, identify not only crime, if some accident happened and you're not able to identify the person by his body or face or something, then we have to go for a DNA matching, something like that you have to do. So that's about the second video on 21 BE 45 biology for engineers, which is common to all four semesters. Second topic, that is nucleic acid. If you need the class note in PDF form, you can contact me on my phone number, WhatsApp number 797-505-4865. I will charge 1 rupee for one class note. Previous note it's already there in the right is running. Second note, I will give this note this much, 1 to 6 pages, and you have to charge. This is 1 to 4 pages, sorry. You have to charge, you have to pay 1 rupee to me. I will not give anything free. Whatever given free, that is not working. That's why. So thank you very much for listening to my YouTube channel, my intuition. Please do subscribe to my YouTube channel and circulate this video to others, those are in the fourth semester of VTU Karnataka. Thank you very much.